Mead Steel Education Productions and Rockingham Studios present Wild World of Science Biology 101 Cell Cycle Review with course instructor Mr. Ronald Jowsey.
Greetings one and all. Welcome back to Biology 101. Today we are going to review what we've learned in class by discussing and taking important notes about the cell cycle. To complete this assignment, you will use the information given in this video to update your class lab notebook. Press pause or rewind the video as needed to complete your notes. Let's get started, shall we? To remember the stages or phases of the cell cycle, I like to use the phrase, "I pretend my aardvark talks crazy." The first letters in each word in this sentence can be used to remember the phases of the cell cycle. These are interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. The part of the cell cycle that is mitosis, the division of the nucleus, is prophase, metaphase, anaphase, and telophase, or PMAT. I can remind myself that the function of mitosis in multicellular organisms is to make body cells for the growth of the organism and the repair of damaged body tissue by thinking of the phrase, "Mitose is part of my body." This reminds me that mitosis makes body cells. Let us now take a closer look at each of the stages of the cell cycle, beginning with the period of time that the cell is being a cell and carrying out its functions. This is interphase. Interphase is that period of time in the cell cycle when the cell is carrying out its normal functions. The cell is also growing and replicating its DNA and other organelles in preparation for cell division or mitosis. During interphase, the DNA exists as chromatin, and chromosomes cannot yet be seen. It is important to note that while chromosomes cannot be seen during interphase, they do get replicated during this stage of the cell cycle. A replicated chromosome consists of a chromatid and a sister chromatid, joined together at a structure called the centromere. Remembering I pretend my aardvark talks crazy, we know that the next phase of the cell cycle is prophase. Prophase is the first stage of mitosis, division of the nucleus. During prophase, the double-stranded chromosomes become visible. The nuclear membrane surrounding the chromosomes begins to disappear. Centrioles are on opposite sides of the cell, and the spindle fibers attach to the centromere of each double chromosome. Near the end of prophase, the spindle fibers pull on the chromosomes and move them towards the middle equator of the cell. From prophase, mitosis proceeds to metaphase. Metaphase is the middle phase where the double-stranded chromosomes line up along the cell equator, the middle of the cell. Once the double chromosomes are lined up down the middle of the cell, anaphase begins. Anaphase is the away phase. The double-stranded chromosomes separate, and the spindle fibers pull the individual chromosomes towards opposite poles, or sides, of the cell. With anaphase complete, and individual chromosomes at opposite poles of the cell, the final phase of mitosis, division of the nucleus, telophase begins. There is now a complete set of chromosomes on each side of the cell. A nuclear membrane forms around each set of chromosomes, creating two new nuclei. The cytoplasm begins to pinch off, and the chromosomes unwind and return to the chromatin state. The final step in division of the cell is cytokinesis. Cytokinesis is the division of the cytoplasm. The pinching in of the cytoplasm that begins during late telophase continues during cytokinesis, resulting in two new genetically identical daughter cells. It is important to note that cytokinesis in plant cells differs slightly than cytokinesis in animal cells. In plant cells, the formation of a cell plate divides the cytoplasm and becomes the cell walls of the two new daughter plant cells. That's a wrap on your cell cycle review assignment. Review the material as necessary and turn in your lab notebook on test day. Until next time, be safe, make good choices, and I'll see you soon.